Okay, I want to talk about the differences and similarities between mitosis and meiosis. Overall, the goal of meiosis is to form 4 genetically varied haploid cells. It's going to be our gametes, our sex cells. So that occurs only in the ovaries and the testes in animals. This also happens in plants as well, but in different cell types, or organ types there. The goal of mitosis is to form 2 genetically identical daughter cells, diploid, I should say, if we start diploid. Excuse me. Genetically identical diploid cells. If we start haploid, it would be haploid. It's just going diploid to diploid, or could be haploid. We'll see in plants to haploid. On the other hand, meiosis is diploid to haploid. That's the point, one of the points. All right. And this process of mitosis is happening in what we say called somatic cells, body cells. So if we start with the same beginning, nuclear envelope. This is a diploid organism. We're going to have 4 chromosomes. 2 are going to be big chromosomes. And 2 are going to be little chromosomes. So they have no, they're decondensed at this point. They are not associating with each other or anything like that. They're inside the nuclear envelope. So they'd be, I've already drawn an S phase. Sorry about that. Before that, let me just sketch really simply black. Without a nuclear envelope, but prior to that, in the nucleus, it would look like this. I'm doing the same for both of them. Okay, so, right, that would be, we'll draw the nucleus, inside the nucleus. So this would be S phase. And this would be something like it's in G2 and interphase, okay? So it's ready to go. It's duplicated its centrosomes, and it's going to divide now. In the end, we're going to, in this case, in mitosis, we're going to get a cell that looks exactly like this cell, 2 of them. In mitosis, we're going to get a cell that looks like this, but with only 1 big and 1 little. 4 of them. So this one, you'll notice, is going to be exactly the same. And that's going to be different from meiosis. Okay. The first stage of all these processes is a prophase. So in meiosis, we have 2 rounds of division. So we call the first round 1 and the second round 2, Roman numerals. So prophase 1 is the first part you go through in meiosis versus just plain old prophase of mitosis. Sorry. There we go. So no numbers there. So in prophase, and we'll call it prometa as well, because we're just going to compare these 2. Okay, nuclear envelope starts to break up. We have our centrosomes that have moved. They're moving around the spindle poles, or moving to the poles, forming a spindle. We have chromosomes condensing. Maybe chromosomes condensed so we can see them better. They are replicated. Oops. Okay, in prophase 1, we have a scenario that's different than prophase of mitosis. In prophase 1, we have our chromosomes pairing with what we call their homologue. So they're pairing with a chromosome that has the same genes on it in the same order, maybe different alleles, but still would have genes A through Z, or not that, we'll say A through D on these two chromosomes, and E through G on these two chromosomes. So they are pairing with their homologous partner. You'll notice here, finish drawing, there are homologs. It is incorrect to say that there are no homologs in mitosis, because there are. They just don't care about each other. They're not going to be involved in pairing or anything like that. So homologs are present, and we know that has to be true because we're diploid. So if you're diploid, you have to have homologs. But there is no synapsis. So this is what's called synapsis, which is this pairing of the homologs. And after we do synapsis, the cells are going to be able to switch little parts of their DNA, and that's going to allow for genetic variation to occur. Okay, there we go. So this is going to be late prophase 1. Okay. Late prophase 1, we've lost the nuclear envelope. I'll just do 2 of these. We've lost the nuclear envelope, and our paired, our chromatids have switched spots. So I'm going to do this by erasing so you can see. So what happens is these 2 places are going to switch. So at this crossover point, what's going to happen is that on this end of the chromosome, it's going to switch over part from the other chromosome. Do the same over here. So now you're going to have some of the light orange on the dark orange, and some of the dark orange on the light orange. So these are the synapses, these little crossing parts. And the same thing will happen here with our blue and our magenta. Oh, sorry, let me, there we go, blue and magenta crossing over. Okay, so we have that. Oh, there it is. And so that's prophase. They're going to stay together with their crossed over parts and be attached to these spindle poles. So let's actually draw this one here. And they're lining up with their homolog, still in synapsis, or still crossed over at least. They have those little bits that have the other ends on them. It would actually be at the crossover point. Okay, there we go. Okay, so you can see the centrosome is attaching its microtubule to the kinetochore of one of the homologs. Same thing is going to happen these guys. And so here's our centrosome sending out microtubule spindles attaching at the kinetochore, the centromere region of the homologs. So if we write out the important points going on in prophase, the synapsis, this would be, we've shown here actually, that crossing over occurs. That's going to generate genetic variation because previously on the chromosome, it could have been that the red or the dark orange chromosome had a big A and the light orange had a little a. The dark orange had a big B down here and the light orange had a little b. If you switch the ends up, see how bigs are associated and littles are associated? If you switch the ends up, perhaps now the little and the big would now be associated. So this would be on the orange chromosome here, and this would be on the, or the dark orange and the light orange. This would be a big A. So you switch them up, as you switch little ends. Okay. So that's genetic variation for you. That's one really important thing of mitosis, one of our big goals. So that's occurring in prophase. This is late prophase, and really we've gotten to, at this point, because I lined them up, we're actually lined up with the metaphase plate. So this is going to be metaphase 1 once we get lined up there. And the crossing over, you'll want to know it's the non-sister chromatids of the homologs. Let me say that. It's the non-sister chromatids of the homologs, or the homologous pair. When we look at metaphase, just do prometaphase, late metaphase, we're going to have a different scenario. Like I said, the homologs are still here. They're just not interacting with one another. So we're attaching the spindles versus, so the spindle, or the homologs, are attached to opposite spindle poles. Now it's going to be the sister chromatids that are attached to the opposite spindle poles. You'll remember this from before, our other chapter. They're not associating with their particular homolog, they just line up. And so where we said that this is lining up here, it's going to be double file with your homolog. Or we could say as a homologous pair. It's probably a little more technical. Here we line up single file. Homologous pair or homologs are there, but they just don't pay attention. So now we're in metaphase. Okay. Then you'll notice also that here I've drawn it to where the dark orange and the blue are on the same side, the light orange and the pink. But there's really no reason we couldn't have, and I'm not going to draw the crossed over parts, but there's no reason it couldn't have been lined up like this. There's no reason that that wouldn't have happened. And so you can see that once you separate, you're going to see these chromosomes and these chromosomes go together. And so in the end, you get two combinations. Over here, and we'll see this in a minute more technically, you'd get these pairings. While down here, you get these pairings. And you'll notice that the chromosomes are not with the same partner, which means they're not with the same alleles, the gene types, the versions of the genes necessarily. Okay, so this event here of lining up, and you'll notice that the littles and the bigs don't influence each other's lining up. That's what we call independent assortment. That's in meta 1. It means they can line up, and I'll do it with genes, because that's really what we're looking at. So it could line up with genes like this. Do the same orientation. Could line up with its bigs together and its littles together, or it could line up with mix. And you see that when it splits, it would be bigs and littles, or when it splits, it would be mixed there. And so that's all valuable options. They should all be, it should equally split between lining up one way and lining up another way. So that's random, generating variation because you're putting different combinations together. In Anaphase 1, that's basically what we just showed with the splitting. Actually, looks like we're doing it already. I didn't want that. Let's make it look bigger. Okay, they're still crossed over. Now we're going to split. So you see that. So it's going to get longer. I'm going to pull the orange X, and I will orient it. Oops. Remember, we have little bits of the homologs on the other chromosomes because we did crossing over. This is pulled by the spindle, of course, to opposite sides. Okay, that's Anaphase 1. Segregation of alleles, meaning that the big A that's on here and the little a that's on there are going to be separating to two different cells, and the B's on this one as well. So now we only get one A in one cell and one A in the other, one B and one B in the other, because that's what we're looking for. We're looking for having the amount of DNA getting haploid. We're still diploid. 2n equals 4. Same thing here. Okay. Same thing here. Because you have two bigs and two littles. Okay. If we go to anaphase of mitosis, we have a little different scenario. No mixes, actually splitting the chromosomes at their centromeres, their centromeres. Okay. Okay. So, in anaphase, we separate homologs. In anaphase 1 of anaphase 1 is enough. In anaphase of mitosis, we separate the sister chromatids. Okay. So that's an important difference there. We're still diploid. Okay. Then you go to telophase 1. We call telophase of mitosis. Nuclear envelope starts to reform. Cytokinesis starts to occur. This must be an animal cell because I've drawn it where it has a cleavage furrow going on. Okay. Here we have one big in each new forming cell and one little. Okay, one big and one little. If the cell, the whole thing is technically still diploid, but each little part that's forming is now haploid. Each new nucleus is haploid. It depends how it's phrased. If they talk about each new forming cell, each of those is haploid. Here, chromosomes are decondensing. We have two diploid cells being formed. You can imagine after that, we get two separate cells with a formed nucleus. You see they're going to have a single copy of each chromosome and two of each type. So it has its homologs still. So it has two bigs and two littles. Same genes, possibly different versions. Okay. So now we have two diploid cells, genetically identical. Mitosis is finished. Meiosis, on the other hand, has two genetically varied cells, not only because of crossing over, but also because they are now haploid and only have parts of the DNA. So we have one haploid cell, genetic variation. We have the other haploid cell with genetic variation. Very important. So after meiosis 1, this is key, you're haploid after meiosis 1. You stay haploid after meiosis 2 as well. But you're haploid after the first division. The problem is that you have two copies of every chromosome. You don't want that. And so what's going to need to occur is you need to break those apart. And so that's meiosis 2. It's very similar to mitosis. I'm going to draw it just across. Each cell loses its nuclear envelope again. Now the chromosomes are going to line up in the middle, just like they do in mitosis. I should probably draw these sideways. These are going to split the opposite direction, turns out. Okay. We have our spindle coming in from here. Attaching. We have more spindles. So this is mitosis, excuse me, not mitosis. This is prophase 1, no crossing over because there are no homologs. Prophase 1. Then we've gotten to metaphase 1, once the nuclear envelope's gone and they're lined up. The only problem here is that they're going to line up and pull. You see, this is a problem because we want them. No, this is fine, actually. Yeah, we're going to pull our sister chromatids apart. Good. Okay, so we're pulling our sister chromatids apart. I guess we skipped anaphase, so let's go to anaphase first. Anaphase 2 specifically. So remember, this is all haploid, all this process. So we're going to split the sister chromatids just like we did in mitosis. But now you see that they're genetically varied and haploid. There we go. Okay, and then we split our little ones as well. So that's your anaphase 2, and then what you're going to get is going to telophase. This first, nuclear envelope reforms, and let me just draw the final cells for you, so you can imagine that. It's actually 4 of them. I need more room. Okay. So each cell has one big chromosome. sometimes there's crossed over parts, sometimes there might not be. And each one has one little chromosome. Again, sometimes there might be crossed over parts, sometimes not. They're not crossed over here, they're just draw that the opposite, actually. Sorry. They are just decondensing. So I want these to be together. So I need to draw blue there. Okay, with a little bit of pink on it. A straight blue one over here. And then from the top, a straight pink one. And then a pink with a little blue. There we go. Now we separated it correctly. So you see this went to these two cells, and this guy went to these two cells. Okay, so now we have 4 different gametes. So you can see that they are indeed genetically varied. They are haploid, only one big chromosome and one little chromosome. They are also, there's 4 of them, and they'll be ready to go undergo fertilization to form a new zygote. You've noticed that this is very similar, lining up wise, to mitosis, except for its haploid and the genetic variation. But we separate sister chromatids. In Anaphase 2, we attach spindle poles to attach sister chromatids, that's how we've been saying it, to opposite spindle poles and then we pull them apart. And then we end up, after cytokinesis, with 4. Okay? So that's going to be important similarities and differences between mitosis and meiosis.